Greetings and welcome to our fourth and final lecture, at least with me, for the head unit. You guys will still go into the eye and the cranial nerve anatomy with other instructors, but we will finish off with the brain, brain vasculature, and their ventricular system. So these are topics I actually find the most interesting in this unit. Brain's just kind of cool all around, in my opinion, so let's get right into it. So here are your objectives for this unit. This will help you focus on where you should study. Again, if you have any questions, feel free to reach out. All right, so we're going to go into kind of a general neuroanatomy for this course. It won't be as detailed as what you might get in some of the other courses because a lot of you will probably actually have a neuroscience course where we'll get into the names of all the sulci and gyri and all that fun stuff. But if you can look at the big picture, that's kind of the goal for our anatomy experience. So let's go ahead and start with what we're looking at here. So this is the brain, and more specifically, we're looking at the cerebral part of the brain. So this is kind of our higher function. A couple of things that you'll see here is this side has all its veins intact. This side does not, so it looks like most of the pia mater is probably moved over here. So remember we talked about that. In the middle, we have this nice long longitudinal fissure. And so if we remember, our falx cerebri is what's going to go through that. So above here will be our superior sagittal sinus. The anterior part contains our frontal pole with our frontal lobe, and we'll look at the lobes in a little more detail in a second. And then we have our back, our occipital pole. And so anytime we talk about the front of the brain, we're usually talking about frontal, and the back is occipital. So let's talk about the cerebrum. So this was the largest part of the brain, and this is kind of unique to most mammals and the higher thinking individuals around in the animal kingdom. And the bigger your cerebrum is, usually the more higher thinking your functions are. And so the cortex, as it's often called, is comprised of gray matter and white matter, and we saw that in the spinal cord. However, it's a little flipped in the brain. So in this case, instead of the gray matter being on the inside, in the brain, it's actually on the outside, and the white matter is actually on the inside. So if you're taking a look at a cross section, you'll actually see the white matter kind of being more centrally located. So just remember it's flipped from your spinal cord, gray and white matter. So remember, gray matter contains the neurons, white matter contains the axons transmitting those signals. So all the neurons tend to be located on the outside of the cerebrum. So we do have two hemispheres. We have a left and right hemispheres separated by that longitudinal fissure there. And then we also have the cerebellum, which is going to be underneath, and that's going to be separated by those dural reflections we talked about earlier. So we have the cerebellar tentorium and the falx cerebri. So those are going to actually be in place. And you can't really see the tentorium cerebelli on this image, but the falx cerebri is going to separate the left and right half, and then the back portion of the brain, the occipital region, is going to be sitting on top of that cerebellar tentorium. So getting into the lobes and kind of what they do. So again, we're thinking big picture here. So the frontal lobe is going to be located from that frontal pole we mentioned to the central sulcus. And you can see the central sulcus here. It's usually a prominent sulcus on the brain. And all brains, even though it looks like it's kind of chaotic in these sulci, and so the indents are called sulci, and these prominent features, these folded areas, are called gyri. All brains tend to have a central sulcus that's pretty easy to identify here, and that marks the frontal lobe. So going from that point forward is our frontal lobe, and that's going to be a lot of our personality, a lot of our higher function thinking occurs in the frontal lobe. And so if you're familiar with the story of Phineas Gage, he was a railroad worker. Try saying that 3 times fast, apparently. Who got a railroad spike up through his orbit into his frontal lobe. And he was always described as a nice gentleman, but then after that accident, his personality kind of switched around, and he became a little more aggressive and mean. And that was because of his frontal lobe being damaged. It kind of inhibited some of his functions and things like his neurological inhibitions. Interestingly enough, our frontal lobes don't usually develop till we're in our late 20s, and that's why we make all our bad decisions in our late teens and early 20s, because our behavioral center is not fully developed. The other lobe is the parietal lobe, so that comes right afterwards. So if we get to that central sulcus and start moving back to our parietal occipital sulcus, that's going to be our parietal lobe, and parietal lobes is pretty much for sensory integration, so kind of our proprioception, figuring out our space in the world. So if there's any sort of lesion there, we can start losing our grasp of like reality in a way. We can't, like, we'll start tripping over things, we'll lose our ability to just coordinate ourselves. The occipital lobe is going to be everything in the posterior aspect here, and that's predominantly for vision. So all our visionary neurons are coming from the occipital lobe. Oops, going a little bit too far. And then finally our temporal lobe. So this is of the four main ones. This lies below the sylvian or horizontal fissure. So that's that like fissure. So if you were to kind of use your thumb or your hand and thumb as a mock brain, this would be my temporal lobe. There's that sylvian fissure, and this is my remainder of the cerebellum, or sorry, cerebrum. And that's where the temporal lobe is going to be. And the temporal lobe is kind of helps kind of keep us alert. And so anytime you get tumors here, this is when you can start feeling like hallucinating and feeling like you're not really awake or part of the world. That all tends to happen in the temporal lobe. We'll also see it actually has important roles in speech manifestation and hearing, and as well as tied to our memories going forward too. So anytime you get dysfunction there, maybe a stroke or some sort of ischemia, we start having issues with things like speech and just having issues with our memories and things like that. So here's a nice little diagram where you can kind of pick out the individual lobes. And again, like I said, for simplicity's sake, we're mainly worried about these four lobes. But looking at it from above, you got that longitudinal fissure dividing left from right. And you can see where that central sulcus is dividing parietal lobe from frontal lobe. Okay. And then here, they don't really have a good, the parietal occipital sulcus is not as prominent as the central sulcus. So you just kind of arbitrarily assign it here in this case. But usually you can see some sort of semi-sulcus forming in that back region. And as soon as you go to the occipital pole, you know you're actually at the occipital lobe for that. So functional areas of the brain. So everything in the precentral sulcus or precentral gyri is motor. Everything in the postcentral gyri is sensory. So what does this mean? So that central sulcus is a nice dividing point. And all our motor, I find this part of the brain fascinating. So we call this the homunculus. And so what happens is all our motor control for everything we do in the body is pretty much localized along this gyrus. And our sensation is located in the parietal lobe under the postcentral gyrus. So anytime you get a sensation or you're receiving sensory information from your environment, it's relaying to this gyri. And the location that it relays is corresponding to this homunculus. And I always thought these are just weird images, particularly this one. This kind of freaks me out a little bit. But it's just kind of a visual representation of how much of your brain is devoted to those particular regions. So here you can see, this is a coronal section. We've got our longitudinal fissure kind of going down. And then we see that our lower extremity is kind of controlled in the top part of the brain. We have arm control in the mid part. Our speech is closer to our temporal lobe region. So that makes sense. We'll see where that plays a role going forward. And then sensory is kind of the same thing, similar function. So that means if we lose function in some of these areas, it has a direct impact on that correlating spot in the rest of the body. So when we do see things like strokes in patients like losing blood supply to very specific areas of the brain, it's a direct correlation. So if we were to somehow lose, get ischemia here, that means the patient could lose motor control if it's from the postcentral gyri, motor control to their extremities, or they can lose sensation if it's in the, post, that would have been precentral, this is postcentral. So the homunculus is kind of a nifty little thing. A couple of things I already kind of alluded to. So there's Wernicke's area and the Broca's area. Those are very prominent in speech, and being able to understand and communicate speech. We won't go into the specifics of those regions, but just know that Broca's tends to be in speech manifestation and Wernicke's is kind of speech understanding. And when we get those areas affected by a stroke, we can see how that can impact it. And we have that typical classic appearance of a stroke patient with difficulty speaking and having trouble kind of in their motor control in that central area. And we'll get to the end of the lecture and talk about that in a little bit more detail. So there's our homunculus. If we look at the inferior, so you guys will go into more details with the cranial nerves, but you can kind of see the cranial nerves we started alluding to in the earlier lectures. So we have our olfactory bulbs. These are these little tiny, olfactory nerves with bulbs attached. These are these little tiny structures, kind of flimsy, hanging on the inferior aspect towards the frontal lobe. See how they kind of ride along the longitudinal fissure? You can actually see the optic tract and chiasm. So that's like this little V. And you guys will see how that plays a role with vision and why the tracks cross. And then you can kind of see some of the other cranial nerves that we talked about here. We won't spend too much time on them, but this is kind of a nice overview of the brain. Here we got our frontal lobe. There's our cerebellum. There's our temporal lobe. You can see our pons here is the prominent structure on our brainstem. And then you can see how the rest of our brainstem leads eventually to our spinal cord. So going from the medulla just underneath the pons all the way to the spinal cord, going through that foramen magnum. So that's an inferior view. You guys will get comfortable looking at that in your lab guides, as well as just getting your cranial nerve review lecture. So let's go ahead and look at the parts of the brain that aren't the cerebrum. So it shouldn't be easy to get those mixed up, but sometimes I do. But so we looked at the cerebrum, the higher function. Now let's look at kind of the basic function of the brains, or the functional areas of the brain. So the one area that we'll talk about is the diencephalon, and that's kind of the control center of our brain. So the diencephalon is made up of the epithalamus, the thalamus, and the hypothalamus. It also surrounds the third ventricle, which we'll talk about in a little bit when we get into the ventricular system. But the epithalamus is kind of a control center. So I'll go ahead and pull up the image here. So this region, this orangey part, is our diencephalon region. And I always like to look at it, especially in this image of the brain, for some reason it always just makes me think of like a mad scientist, and this is this like crazy hat, like maybe his doomsday hat or something. He's got a little eye and face. So I don't know, it looks like a face to me. I could be crazy. But the epithalamus is this little body back here with our pineal gland, or pineal gland, depending on how you want to say it, is back there. And that helps control some of our circadian rhythms and sexual maturity. So being able to stay awake and fall asleep on a proper schedule is all done in the epithalamus. The thalamus is really the main relay center. So everything will eventually come to the thalamus and then be organized out. And that contains what the structure is known as our basal ganglia. And these are like these fine motor controls that kind of exist in this region. And so the thalamus is really just the biggest portion of this. And then you have the hypothalamus, which is right here. And that's kind of connected to a lot of autonomic functions, and it actually has association with our pituitary gland. But ultimately, a lot of our homeostasis is maintained through that hypothalamus here. And it looks kind of like, I don't know, the lips of the old man, if I go back to that analogy. And this is his face. So the big part is the thalamus. We got epithalamus over here and hypothalamus in the front. Okay, now brainstem, if we go back to that image, the stuff in yellow is our brainstem. Okay, and we already saw the pons is that prominent structure there, followed by medulla underneath, but let's go ahead and take a look at the structures more specifically. So the brainstem is made up of the midbrain, which is pretty much in the middle of the brain, the pons, and the medulla. So the midbrain, which is pretty much the junction between our diencephalon and our pons. So that area right here is our midbrain. So this is going to be a lot of our audio-visual control. So we see a lot of relays between our audio and our visual portions of the brain. So remember, our occipital is actually going to be in the back of the head, so we get a lot of control over there. We also have pain modulation, and a lot of our motor control is going to be through there too. There's an area called the substantia nigra. It's part of the basal ganglia, so again, it takes that relay information and helps kind of control our motor movements a little bit more refined. And it also plays an important role in Parkinson's disease, in that when that area becomes deficient, that's when people have symptoms of Parkinson's, and that's where you kind of have like the uncontrolled pill roller movement, they call it. But again, you don't need to know about the basal ganglias, just know that they're kind of located in that midbrain region. Next is the pons. So the pons is that big structure here, kind of more prominent on the anterior aspect of the brain, or the ventral aspect of the brain. So our pons is a very characteristic structure on the brainstem. It helps with a couple of other like non-specific, what should I say, basically just some homeostatic type things, a lot of non-controlled movements. So things we don't think about, I suppose, the autonomic stuff of our body. So things like eye movement, salivation, crying, facial muscle movement, just things that kind of are reactive components all kind of go through our pons and help relay that information as well. So if we do have general issues with just overall maintaining the state of our body, a lot of times that could be issues with our pons, especially you can see some hemorrhage there when we get in some of the pathologies. And then finally the medulla, or medulla oblongata for the full name. And so that is basically just the last portion before we get to the spinal cord, and that exists right about here. So that's the final portion of the brainstem. And what that is really just going to be a lot of our, again, more internal structure maintenance, stuff that we don't generally think of, things like breathing, digestion, just your basic function of your non-controlled, basically your smooth muscle organs in the body are kind of regulated by your medulla. The biggest one being things like your diaphragm and breathing, respiration. And so if you remember I mentioned when we talked about hernias, this is the area that can basically be pinched off in that foramen magnum and cause a severe lack of function because we lose our ability to basically regulate breathing. And that's something we shouldn't be thinking about. Breathing just kind of happens, and it all happens inside that medulla down there. So here's kind of our overview picture. So again, we have the cerebrum, we have our diencephalon, which are thalamus. We don't really see the epi or hypothalamus. And then we have our brainstem here with midbrain, pons, and medulla following these mid structures. So then the final thing is that little tree-like structure in the back. And I say tree-like, but it actually is referred to as arbor vitae, which is tree of life. I always thought that was kind of nice and poetic. But this is our cerebellum. And so this is a lot of fine control. So think of this as your athletics portion of the brain. So rather than just doing big bulky movements and just like, oh, I want food, grab, eat, the cerebellum lets us do a lot of things like control, balance, standing on one foot, being able to stretch and run and maintain all that flexibility to the point where if there are lesions of the cerebellum, you experience things like ataxia, which is sort of a loss of fine control. So this is, the cerebellum is prominent for a lot of those limb coordinated movements, I should say. So this is just kind of a summary of everything we talked about. So you can basically just take that and then study it off of this slide, and it should just be as good. All right, so let's take a look at the ventricular system in the brain. And the ventricular system is a way for the cerebral spinal fluid to kind of keep regulating itself. So I said in an earlier lecture when we were talking about the meninges that the cerebral spinal fluid is the equivalent of lymphatics in the brain. And this is how the system kind of starts. So during embryological development, we have what's known as a neural tube. So it's just a singular tube that grows and it kind of folds in on itself. So it's got a hollow center. And what happens is eventually that tube's going to keep growing and partitioning and folding in on itself so that the hollow part on the inside stays hollow. And that's basically what becomes our ventricular system while everything else continues to grow and expand and become the rest of our brain. And we see that basically the hollowed portion maintains all the way through to the spinal cord. So there's actually the central canal in the spinal cord that is continuation of our ventricular system. So in the brain, we have essentially 4 ventricles. We start with the lateral ventricle, and we don't ever really call them ventricles 1 and 2, but they are in essence ventricles 1 and 2. For identification purposes, just call them lateral ventricle. You don't usually have to say left or right unless specifically asked. But these are going to be these large C-shaped structures on the edge here. So this is going to be kind of in the midpoint of your cerebrum. They're going to be separated again by that longitudinal fissure, so you have a left side and a right side. But these C-shaped structures are your lateral ventricles. They're the biggest ventricles in the brain, and this is kind of where everything starts. So you basically have these ependymal cells in the ventricles that are going to create this CSF and start building it into the lateral ventricles, and those are eventually going to fly or follow out into the third ventricle via this interventricle foramen, which is not labeled here, but you can basically see is, oh, there it is. It actually is labeled. Sorry, I'm trying to get my point. Right there before it gets to the third ventricle, which is going to be contained within our thalamus that we mentioned earlier. So that big circular thing is our third ventricle. So notice we have lateral ventricles and it just skips right to the third ventricle. So then things are going to collect in the third ventricle. And remember, this is going to be kind of located, our thalamus is around the third ventricle. And then everything's going to coalesce into the fourth ventricle and travel by the cerebral aqueduct. And so that you can see here till we get to our fourth ventricle right down here. That's going to be just directly anterior or ventral to our cerebellum. Okay, so the lateral ventricle, everything kind of gets in, forms, goes through the cerebral aqueduct, the intervertebral foramen into the third ventricle, through the cerebral aqueduct into the fourth ventricle. And that's essentially our ventricle system. That's everything kind of getting contained within the brain. And eventually that's going to make its way out into the subarachnoid space that we looked at earlier. And the way that happens is through these apertures. So once we get to the fourth ventricle, basically everything's going to dump out into one of three possible apertures, which are just openings into the subarachnoid space. You have a median aperture, or a foramen of Magdeni, or Mag-ND, however you want to pronounce that, and a foramen of Luska, which is your lateral ventricle. So you have two lateral ventricles and one median ventricle. Remember, it's not medial ventricle or medial. It's not median. It's not medial aperture. It's median aperture, and it's lateral apertures. Okay, so you can kind of see those here. The median aperture is coming right on the posterior aspect, where our laterals kind of go anterior. You can see them a little better here, actually. So there's our median, and our two laterals. We're basically going to be becoming more ventral. So here's our nice coronal view. So here you can see, and this is what we were talking about earlier. So all this, I guess you call it brown, is our gray matter, and then the white matter is on the inside. We have our lateral ventricles. We have our third ventricle. We don't see our interventricular foramen, but we do see our cerebral aqueduct, and then we see our fourth ventricle down here. And then it'll continue on to our central canal. So after we exit those apertures, now we're in the subarachnoid space. And so our cerebrospinal fluid is basically just going to keep surrounding the brain, and then eventually leaves the subarachnoid space through those arachnoid granulations into those venous sinuses that we looked at from the dural reflections. And then basically start the process all over again. So what does the cerebrospinal fluid do? This is basically just going to be a cushion. It's going to get rid of all the waste, the same way that lymphatics would, and basically just keep replenishing the brain and remove all the unwanted debris, all the unwanted hazardous proteins and things like that that get kind of lodged in there. Just kind of cycle them through the brain. I actually do say that part of, like, when we sleep, our brains, they'll like shrink a little bit and like allow for more CSF to kind of go through and rinse them out, and helps kind of clear them out. So if you're kind of sleep deprived, what ends up happening is that stuff kind of sticks around, and it sort of increases the likelihood that you'll suffer from dementia or Alzheimer's, because you basically have this buildup of proteins and things in the brain that shouldn't be there, and then it should get cleaned out. So if you ever need an excuse to sleep, I think that's as good as any. So sleep away. But here's just some facts. You can see that it turns over quite frequently in the day. Basically 600 to 700 cc's or milliliters are produced each day. So here's kind of an outline of the system. So this is a combination of what we talked about just now with the ventricular system, as well as what we were talking about with our subarachnoid space system. So again, we see that the CSF is being produced in the lateral ventricles. It's making its way to the third ventricle, then all the way down to the fourth. It leaves through those three potential apertures, and then it's going to get into the subarachnoid space, going to float around the brain, and eventually through these granulations, it's going to make its way back into the venous supply system. So there are these places called subarachnoid cisterns. Really, this is just areas of enlarged subarachnoid space. So you can kind of see them. You can see them a little bit in here, but I like this image a little bit better. Plus, they're labeled. So the most prominent one is this posterior cerebral medullary cistern. It's kind of on the dorsal aspect of our brainstem, and so basically it's just a prominent dilation. So normally our subarachnoid space is pretty consistent and relatively small. This is obviously drawn a little bit bigger for artistic embellishment, but this cistern here is going to be probably one of the bigger ones we see. We also see, well, they call it the pontine cerebral cistern over here, but that's actually just pontine cistern is also an identification for it. So that corresponds close to where our lateral apertures are coming out. So you can kind of see that, let's see here, right around here. And then You also get your interpeduncular cistern, which is going to be up by the pedicles, which we don't really talk about in this class. They're just going to be more of that relay from the midbrain, but those will be up by, missing it right over here. So here's where an easy way to find that is this structure is known as a mammillary body. Again, nothing that you're going to be tested on, but the cistern is located right there by the pedicles. So. All right, so now taking into account everything we've already looked at, this is kind of a nice representation of all their spaces in the brain. So we looked at venous sinuses before when we were talking about dural reflections, and so the main ones were sagittal sinus. So remember, we have a superior and inferior one. We can't see the inferior in this particular section. We have the cavernosus sinus, which is going to be that anterior aspect right around here. We also have the transverse sinus, which are going to be those lateral sinuses right around that cerebellar tentorium. And then we have the sigmoid sinus, which is going to be this guy. So actually call that transverse. This would be like our cavernous. The confluence of sinuses is going to be in the back, and then we have our sigmoid, which is going to make that little loop before it ultimately goes into the internal jugular. And then up here you see in green is our ventricular system. So we have our C loops coming into our third ventricle, and we can't see the cerebral aqueduct or fourth ventricle on this image. Here you can see it much better though. And again, this is just showing the relationships between those various spatial systems in the brain, so you can kind of visualize where they're actually located and how they function among themselves. All right, so let's go ahead and take a look at some clinical correlations and specifically pathologies for the cerebrospinal fluid. So this is something that you can see in children, and we already talked about the fontanelles and how something like impinged drainage of your cerebrospinal fluid could lead to an enlargement of the skull. Now that's not to say you can't get obstruction of your ventricular system in an adult. The problem with there is that your brain is already well developed and formed, your skull is hard, and so anytime you get any sort of increase in volume in the brain as an adult, you manage to see those symptoms quite early. Usually manifests as a headache. There's a lot of buildup of pressure. It's that mass effect issue that we mentioned where stuff's going to start pushing on those dural reflections and moving to places it shouldn't be. Now in children, and basically those fontanelles haven't developed entirely, so the brain is just basically adapting with the skulls and the fontanelles adapting to that change. There's two types to be mindful of. You can have obstructive hydrocephalus, which is where you basically are losing some sort of blockage in the ventricular system. And then there's also communicating, where your arachnoid granulations basically are impinged, and so the cerebrospinal fluid is building up in the subarachnoid space, and it's not being allowed to leave that area into this the sinus system. Obstruction tends to be a little more common, and that's what we see with the enlargement of the ventricular system here. But things like tumors can also impinge on that. If the CSF isn't being allowed to leave the ventricular system, you'll see a dilation of those ventricles. Here you can see a really good image here of the dilated lateral ventricles. And so basically what you got to just remember is wherever your blockage is occurring or lack of communication is, the effect is going to be downstream. So if we have blockage or a tumor in our skull base, and basically our fourth ventricle is being impinged, we'll see something like this, where our buildup is going to be in the third and lateral ventricles. Typically these are caught early enough, and even in children, because the brain is a little more amenable to the restrictions of the skull, because the fontanelles are going to allow for growth. Once you shunt the fluid away, it's basically you can repair it, and hopefully with minimal amount of brain damage, because you haven't damaged the neurons specifically, you've just sort of compressed and changed the volume of the brain. What they usually do is they just shunt it into the abdominal cavity, so you can just kind of implant a shunt from your ventricle into your abdominal cavity. The abdominal cavity is much more geared towards handling a lot of fluid or excess fluid than anything else in the body, to a certain degree, because you can get things like ascites, but that's usually the go-to fix. And then we just see some averages or some comparisons between your normal ventricles on a CT versus a dilated ventricle through hydrocephalus. Lumbar puncture, you guys kind of talked about this in the intro unit, just something to be mindful of. Again, your goal is to monitor the CSF and see if there's any signs of infections and things like that, so we won't go into that again. And then also something to be mindful of is brain tumors. So we kind of alluded to these earlier. You can get brain tumors at just about any point in your brain. in really, you can get any type of tumor too. You can get things like lymphoma in the brain. You can get bone tumors in the skulls of the brain. But just for basically simplicity's sake, I'll just bring up these two diseases. So glioblastoma multiforme, or GBM. It's also an astrocytoma grade 4, but you don't have to worry about that. It tends to be the most common primary malignant tumor. And we start playing this little word game with brain tumors. So it's the most primary malignant tumor. And you can see the problem with this is there's really no clear boundaries. In CT scans, we have what's known as an enhancing ring around it. That's a classic sign of GBM. But if you wanted to go and remove this tumor, there's really no easy way to do that. So most brain tumors, especially this GBM, are highly fatal and oftentimes within a year after diagnosis. However, the most common tumor overall is metastasis. So a lot of things ultimately make their way into the brain. That's just the way it goes. The brain takes a lot of our blood supply, so eventually if there are tumor cells in the blood, they'll eventually at some point find their way into the brain. So you see a lot of metastasis in there. So chances are, if you were a betting person and someone had a brain lesion, there's a good chance it's metastasis, but there also could be primary lesions too. But you can get about anything metastasized up there. What's interesting is you don't get the contrary. So brain lesions, because they tend to be self-contained and we don't really have lymphatics other than the CSF, we don't get a lot of brain tumors metastasizing to other parts of the body. So it's always kind of a neat bit of trivia. So now let's dig into the arterial system of the brain. And this is just a nice little artistic rendition of the vessels. You don't have to identify every single one of those, don't worry. So looking at the inferior view, one thing we have in the brain is the circle of Willis, and we'll go into a little more detail of what that is and how to identify those structures. But it's a lot of redundant circulation. So we get most of it from the anterior aspect through our internal carotids. But we also saw that we get the vertebral arteries going up through the cervical vertebrae, those transverse foramen, will come back up and merge posteriorly. So we have we have a sort of redundancy with circulation from the internal carotid and the vertebral, creating this nice little circle between the anterior and posterior aspects of the brain. So ideally, if there's blockage in one area, we should hopefully have it covered. Like I said, the brain takes a lot of our oxygen needs for just our daily life functions. And even when you are going through and doing a lot of study, and studying actually does burn a lot of calories, so you can technically call that a workout. It's just all that oxygen needs and nutrients that our brain needs to keep going on a regular basis. We don't want to deprive it of. So this is a nice highlighted version of the circle of Willis. So I like to start here with the internal carotid. So we're looking on the inferior aspect. So imagine the internal carotid coming out towards you, and it would come up through here and then basically start this. It's almost a direct shot from the internal carotid to the middle cerebral artery. That becomes clinically important for a lot of things like emboli, and we'll see why, because it's so easy for something to come up the internal carotid, and basically it's, rather than take any sort of weird veering, it's just going to keep going straight and find itself in the middle cerebral artery. So then if we go anterior, we have the middle cerebral will be coming up and doing most of the mid portions of the brain. We have the anterior cerebral as a branch coming off of that, going towards the front part of the brain. There's an anterior communicating artery, and this is all symmetrical, so you should see what's on the left is also on the right. So anterior cerebral, this little junction in here is the anterior communicating. We come back here, there's the posterior communicating, which goes from essentially the junction at the internal carotid / middle cerebral all the way to our posterior cerebral artery going towards the back of the brain here. So that's the main stuff that's going to basically supply blood to our cerebrum. We have a couple others to look at here. When we go up, I'm going to start from our bottom. So let's say these two vessels well, not say, but that's what they are. This will be our left and right vertebral arteries are going to eventually come and create our basilar artery, riding up along the ventral aspect of our brainstem here. Okay, a couple of other arteries of note are the anterior inferior cerebral artery, or AICI, and then there's also the PICI, the posterior, which is coming off a little bit, sorry, gotta find my finger, posterior inferior cerebral artery. And so those basically are going to be on either side of the cerebellar. So you'll hear AICI or PICI, Pika, if you're a Pikachu fan, I guess. And so those ones are worth documenting. And then your basilar artery basically connecting up with your posterior cerebral. So those are kind of your main ones that I expect you to know. I'm not too keen on superior cerebellar artery. We don't really look at it too much. The other ones are a little more identifiable per se. So this is another review of what we looked at in the Freeman. So the path of the internal carotid, I told you it was a little unique, mostly because it's usually associated with that foramen lacrum, but that's closed, even though a lot of vessels and nerves are coming through that are trying to make their way. But we actually have a carotid canal making its way up, or the canal doesn't, but the internal carotid makes its way through that canal and kind of like curves over the foramen lacrum before making its way into the circle Willis. So it's a lot of curvature, and we know what's going on in this region is that cavernous sinus, remember? So we have a lot of our main structures in there. So just be mindful of the fact that your internal carotid will go in through the carotid canal on the outside, but you won't see the carotid canal on the internal aspect. Okay, it's going to come up and basically overlay your foramen lacrum when you're looking at the way the internal carotid enters into the brain here. The other thing of note is the ophthalmic artery. So we saw that this was the only superficial facial artery that came from the internal carotid. Everything else was from the external carotid. So one of the first points that comes off before we become the middle meningeal artery, the middle cerebral artery, is this ophthalmic artery. And that's going to basically branch into our supratrochlear and our supraorbital artery as well. So that branch happens before we get into the skull, just as it passes that little curvature region down there. So I just made a complete list of things that you should be the main arteries for this unit. So being able to identify the internal carotid and its branches, and as well as the arteries for the circle of Willis. Really, regardless of ones we already talked about, I'd say just focus on these and their corresponding regions of vascularization. So the ones that say see vascular territories, that has to do with the fact that when we think about the brain, there's sort of a division of labor, right? So we saw where the anterior is coming up towards the front and kind of supplying up here. The middle is actually going to come up through that sylvian fissure and provide the lateral aspect of nutrition, and the posterior is going to do the same for the posterior aspect. What that means is we have specific territories that are covered by our vascular supply. So you can kind of just branch out and think like, okay, so I know that the anterior is going to kind of come up along that longitudinal fissure and cover most of my anterior and probably the superior aspects of my brain. My middle cerebral is going to do a lot of that lateral aspect, and then the posterior is going to do mostly posterior and temporal. So what happens is now these areas create what's known as watershed regions. And so if you think about what that means is basically there's regions of the brain that are getting the watershed runoff. I don't know if that's a good analogy. Sometimes these things are so embedded in weird historical terms that it doesn't make sense. But if you just think about, let's say you fill up a pot or a water bucket to go water some plants, and you always start on the right side of your garden and move to the left. Well, eventually the left always just gets whatever's left over. So ideally you want your hardier plants on that end because you're going to have less water if you're not partitioning out right. So that's kind of what you want to think about in here. So these watershed areas are junctions where it receives blood supply from two different regions. So what happens here is, yeah, our anterior cerebral artery is filling up everything here. Our middle cerebral artery is filling up everything here. But right here, there's kind of a not my job mentality. So if someone drops the ball, it's like, well, if I'm a little lazy or if I have some emboli or thrombi, I'll let ACA do it. But the ACA can only do so much. So when you start dealing with things like global ischemia, and we'll look at that shortly, those areas tend to suffer first. And that's where we see things like stroke patients kind of suffering from those. So again, this is kind of a layout of those vascular territories, just in a more coronal view. But you can see like the areas that are most affected, the watershed areas where there's junctions between the two, tend to be the arm and upper extremity regions of the brain when we think about that homunculus. So here you can just kind of look at the breakdown of those vascular territories and what's being supplied by the anterior, middle, and posterior. Just get comfortable with that. It should make sense corresponding to the regions of the brain that we talked about earlier. And just knowing like, okay, well, the anterior cerebral is going to come up the midline and supply a lot from the sort of superior anterior aspect, and the middle cerebral is going to do a lot of that parietal lobe such stuff. Again, CT scans, I'm not going to be too picky on making you guys know these. There actually is an interesting, I'd have to find the article, but they recently did an MRI, like the highest def MRI scan of a brain. It looks really cool, but they like took hundreds of scans for like hours. I think it was like a 100-hour scan total actually. And then they just make it the most detailed MRI scan of the brain. And if you remember, MRI is really good for brains because the brain is mostly soft tissue. And so a lot of surgeries will use MRI scans for their interventional radiology, whereas CTs doesn't really show you the finer points, but we can see the bone clearly. We just don't see the gray matter versus white matter that we see in the MRI. So hopefully you can really appreciate that difference. Same here. You can see the eyes too, kind of neat and a little creepy. But again, those high definition scans, I'll probably post that in the supplemental area just so you guys can see how cool that looks. Here's a nice sagittal view. Again, notice that you can really, all the structures, I could probably easily ask you to identify the stuff we just talked about. You know, you see your cerebellum, or sorry, cerebrum, cerebellum's down here. You see your diencephalon with your epithalamus, your thalamus and hypothalamus. See all the parts of your brainstem. You can even make out the ventricular system. So MRI images are much better for brains, given the soft tissue nature of them. So let's take a look at some clinical correlates before we call it a day. So we kind of alluded to a bunch of these, but we can kind of just talk about them in a little more detail. So we did talk about things like ischemia and infarcts in the introductory lectures that I gave. Ischemia is any sort of loss or hypoxia, usually local hypoxia, which means a lack of oxygen due to insufficient blood flow. So we get ischemia when there's any sort of blockage. An infarct occurs at the site due to inadequate blood flow, and basically that tissue just undergoes necrosis and dies. When we have a stroke in the brain, that's just going to be a specific area of the brain that basically is dying off because of either ischemia. So you can get global ischemia where maybe there's an overall hypoperfusion of the blood in the brain. Or you can get a hemorrhage, maybe a ruptured vessel. It means like the blood's just kind of pouring out but not going through that capillary system that it needs to go through, and that can cause areas of the brain to die out. So a couple of pathologies that you'll see is a thrombus versus emboli. Thrombus tends to be localized, so you can get a thrombus buildup in a single vessel, but if that thrombus dislodges, it can basically find its way into a smaller vessel and lodge there, and that's when it becomes an emboli. And that emboli can be dangerous because it basically will keep traveling until it basically can't, so it'll lodge itself into a smaller vessel. Whereas a thrombus will start slowly narrowing the lumen until you get less and less blood flow, and eventually leads to hypoxia. Aneurysms we talked about, particularly Barry aneurysms, so that can be a cause of hemorrhagic ischemia or an hemorrhagic infarct in the brain, because basically the vessel will dilate, weaken, and burst, and then it's just kind of leaking out there and not properly providing nutrients to that region. And then the other ones you can get is an AV malformation or an atrial venous malformation, where it's basically just a communication between arterioles and venules without going through a capillary bed. Which means you basically just bypass that system altogether. Nothing's being transmitted, no nutrients are being expelled from the vessels as a result, and that basically just leads to ischemia there. And those usually are congenital defects that they have to go in and actually manually remove from the patient. So a couple of ways that those vascular pathologies can manifest is either through a transient ischemic attack or a stroke. The transient ischemic attack, or a TIA, is really just like a non-specific loss of function, and this could be just anything that basically causes some sort of symptom like a stroke, but it's not permanent, and there's really no evidence of any sort of change in the brain after the fact. So you might see those, just maybe a temporary loss of blood, or maybe some sort of dysfunction, especially if someone is experiencing things like long-standing atherosclerotic plaque buildup, and sometimes you just have a discrepancy in some blood flow that needs to be managed to go in. The long-standing or the long-lasting impact are the strokes. So you can get ischemic, which is usually some sort of blockage of the blood vessel, whether it be a long buildup of thrombus or dislodging of an emboli into the blood supply, or you can get hemorrhagic. So these are less common, but if those aneurysms were to burst, you get a buildup of bleeding in that general area, and that becomes a hemorrhagic stroke. So it has the same symptoms because we're still losing blood to the tissue, even though there's blood surrounding the tissue. So we need that capillary bed, that hydrostatic pressure to kind of get out those nutrients the way we want them to. So here you can see the difference between the two. So in ischemic, you usually just get a sort of dulling of the tissue as it starts to die off because it's just not receiving oxygen, and so it basically walls off. The interesting thing about brain tissue is when it starts to die, we saw in other tissues how it forms scar tissue and becomes fibrotic. So remember in the heart lecture, we looked at a infarct in the heart and it became that nice white fibric scar. In the brain undergoes what's known as liquefactive necrosis. And what happens is when that tissue dies, it just kind of becomes like soup and then gets reabsorbed. So we lose parts of the brain. Compare that to a hemorrhagic infarct, kind of on the way here, we actually see the blood buildup, right? It's just, that's like our hematomas that we looked at earlier. So just be mindful of those two. You can also get global versus localized. So global ischemia is just a general loss of blood supply versus local, which is usually due to the emboli getting a very specific location. So the other thing we'll talk about now is middle cerebral artery syndrome. And so I already kind of pointed out the fact that that internal carotid artery basically has a direct connection to the middle cerebral artery. And what happens is this becomes the most common site for those thrombi, or I should say emboli. So it basically comes up your internal carotid and just goes a straight shot to the middle cerebral artery. And so a lot of strokes tend to be in that general region, which manifests as very similar symptoms. If you think about the areas that are supplied by our middle cerebral artery, especially those transitions or the watershed areas, some of the ones that are most likely to be affected are a lot of these speech areas. So we have things like Broca's or Wernicke's area that will also become subject to ischemia, lose blood, and basically cause the patient to undergo the classic stroke signs of difficulty speaking. So aphasia is a term for any sort of pathology with speech. And so you can either get like a Broca's aphasia where you can comprehend, but you can't speak the words. So you have that inability in the motor control. Whereas Wernicke's is you can say words, you can just talk, but your comprehension and your communication is basically just off. So you might ask someone a question and they'll just start randomly putting together a string of words about whatever words come to mind, but it'll have no sense to it. So their motor is intact, but their cognitive recognition of those words is not. So you need both of those areas intact to form comprehensive speech. And usually you can get one or the other, or in some cases, if it's like a global ischemia, you can lose both of those regions and basically just lose the ability to understand and the ability to speak completely. So that is it for my part at the head unit, and that was brain. Let me know if you have any questions, and thank you for tuning in for these lectures. Take care and have a great day.